Hi, everyone. Welcome and thank you for joining us for our fifth annual NCEAS Spring Seminar Series. Today, we have the pleasure of welcoming Dr. Tanya Berger-Wolf, our first speaker in this series. This year, our series is centered around a theme of leveraging AI for environmental good. I'm Justin Cotty, a data coordinator at the National Center for Ecological Analysis and Synthesis. I'm joined by the rest of the seminar series planning committee, Angie, Erica, Christina, Erica, and Nicole. At NCEAS, we work to accelerate scientific discoveries that enhance our understanding of the world and benefit people and nature, as well as to transform the scientific culture to be more open, efficient, and collaborative. Before we begin, we ask you to join us in acknowledging the Chumash peoples who are the environmental stewards of the land on which NCEAS is located. We pay our respects to the Chumash peoples and their elders, past, present, and future, who call this place their home. We acknowledge that this land on which we live and work are the traditional homelands of indigenous peoples, including the Chumash peoples. This land today is the result of the erasures and exclusions of indigenous peoples, and as we come together to bring awareness to the issues of equity and justice, we recognize that the Chumash people and so many other indigenous people are still fighting to dismantle the legacies of settler colonialism in their tribal territories. As we continue to work towards diversity, equity, inclusion, and justice in data science, we strive to prioritize Indigenous data sovereignty and self-determination. Now I'll introduce you to our speaker for today and give some logistical information about the presentation. I'm delighted to welcome Dr. Tanya Berger-Wolf to our seminar series. For all of you joining us for this webinar, please feel free to add any questions you may have during the talk through the Q&A button located at the bottom of your screen. We will also have a moderated Q&A section including these questions at the end of the presentation. I'll also mention that we have some folks joining us in our lounge to watch your talk today. And now I'd like to introduce our speaker. Dr. Tanya Berger-Wolf is a professor of computer science engineering, electrical and computer engineering, and evolution, ecology, and organismal biology at The Ohio State University, where she is also the director of the Translational Data Analytics Institute. A pioneer in artificial intelligence for ecology, biodiversity, and conservation, she leads the NSF-funded Imageomics Institute and the US-Canada co-funded AI and Biodiversity Change Global Center. She serves as a scientific advisor for many organizations, including the US National Academy's Board on Life Sciences, US National Committee for the International Union of Biological Sciences, the Global Partnership on AI, and the Nature Conservancy. She co-founded the AI for Conservation nonprofit WildMe, now part of the Conservation X Labs, creator of WildBook, and recognized by UNESCO advancing the UN Sustainable Development Goals. Her contributions have earned numerous accolades, including recognition as the AI 100 Global Thought Leaders by H2O AI. Tanya, we're so excited to have you. Please go ahead, and thank you.
Thank you. Thank you for having me, and I am really excited to kind of bring together the concepts of AI for nature. And I often talk about AI for nature from science to impact, but I really want to focus on the trustworthy aspect of AI in this context of what makes it trustworthy. And so with that, to situate this talk a little bit, I'm sure that many of you in the audience, in this particular audience especially, have seen these kinds of headlines that we're losing biodiversity at an unprecedented rate, that over 1 million species are threatened with extinction, and to quantify this 1 million, it's at least 10% of the known biodiversity out there, about 10% of the world's species, which is insanely high number. We're in the middle of what has been termed the sixth mass extinction, and it matters, right? It matters both because where we're losing biodiversity, it is in the places where biodiversity is highest, we're losing it fastest. It also matters why we're losing biodiversity, primarily because of the man-made factors. It's the human activity on Earth that accelerating the loss of biodiversity. And it matters because of what the services that it delivers, right? It's not just nice to have the variety of species out there. Even if you only care about one particular species, us, humans, the biodiversity delivers clean waters, the air that we're breathing, the fertile soil, a variety of medical solutions and treatments, and just the context for human well-being. Yes, nature is not just nice to have, it's actually healthy to have. And so to understand to monitor the biodiversity that's out there, understand its loss, and figure out the interventions to mitigate its loss. Over the last 20 years or so, there has been an explosion of ways of sensing the world, in particularly natural world, from satellite remote sensing and variety of modalities and spectra to autonomous vehicles underwater, on the ground, and in the air, on-body sensor that can measure everything from GPS to an animal's heartbeat, stationary sensors such as motion-activated camera traps and acoustic sensors, environmental DNA, and even these kinds of devices that I'm sure many of you carry. And if this was an interactive talk in person, I would ask how many of you have taken a picture of a living organism in the last week. And I'm sure almost everybody, especially with a reminder that humans are living organisms. So there's tons of data now coming about the natural world from all of these sensors. So one would think, okay, great. So we have data, we've got data. But if we look at the data that is coming about the natural world, then the challenge is that in the top left corner is the biodiversity heat map, terrestrial biodiversity heat map, to be precise, mostly. And as you notice, if you even looking at this map, it doesn't cover the oceans, which is a big source of biodiversity, an increasing source of our understanding of biodiversity. But okay, even with that, if you compare it to the official source of biodiversity, which is the Global Biodiversity Information Facility, which is the bottom left, there is a clear sort of misalignment of where the biodiversity hotspots are versus where we have majority of the data and observations of biodiversity. And the same bias kind of continues for the iNaturalist data, which is the largest citizen science platform, and eBird, which is a citizen science platform and sort of crowdsourced global platform for bird-focused data. So if we look at those maps, one would think that the majority of biodiversity is concentrated in North America and Western Europe, which is obviously not the case. And if we zoom in even more, then the majority of observations are not just close to cities, roads, and parks, nature parks, but they're also the data highly correlated with the average income of the zip code. Per zip code. So this is hugely biased data, and we're lacking most data exactly where the biodiversity is highest and where we're losing it fastest. So the situation is even more so that if we look at the International Union for Conservation of Nature Red List, which is the official source of biodiversity and determination of endangered species and categorization. So the red list is that list where we say if the species are endangered is because the species commission for that species, the red list species commission for that species determined according to some metrics that the species status is endangered. And so if we look at the taxa that are being monitored for biodiversity and critical status in the red list, you already notice the huge bias towards vertebrates, with some invertebrates and plants being monitored and assessed, but fungi is a really, really, really low number that are even being targeted for assessment. But out of all the species, about 160,000 that are being assessed, about more than 22,000 of those, the efficient conservation status is data deficient. So which means that we don't have enough data for any of the metrics to make determination of appropriate conservation category. And another more than 60,000, the population trend is unknown. And so if you're keeping track of the math, this is more than half of all the species that are being monitored and assessed in the red list. They're either data deficient or the population trend is unknown, so which means we're not assessing them frequently enough. And these are not obscure species. Killer whale, the largest dolphin in the waters, hard to miss, orcas. The official conservation status is data deficient. Polar bears, the iconic conservation species, the population trend is unknown. So if we don't have enough data for these species, what hope do mushrooms have? Right. So the very first challenge in mitigating biodiversity loss is knowing what's out there. And so this gaps in our basic knowledge of what's out there is not just that we're losing species faster than we can name them, let alone assess. But this bias that I pointed out is not just taxonomic, that we favor vertebrates over invertebrates, or we, different modalities or different regions, we're biased to different regions or different types of habitats, or for different types of traits or phylogenetic groups. They're different, whatever kind of dimension you're going to choose, there's going to be a bias of our data available in that dimension. And so we'll outline all of these gaps in a recently published Nature paper led by Laura Pollack and Justin Kitsis and a collection of co-authors, most of them from the AI and Biodiversity Change ABC Global Center. And we're also outlining the opportunities where AI can really help not only fill these gaps, but also extend our understanding of biodiversity and ways that we can mitigate biodiversity loss. And the reason that we think that AI is really kind of ready, not just to fill the data gaps and mitigate the biases, kind of align the biases, but also help understand the why we're losing biodiversities. How are these complex factors really interact and contribute to the current state of biodiversity, is because AI really has exploded over the last 10 years, especially. I don't know if you've been paying attention for the last 3 years or so. There's really a different type of AI that's out there. So one would think that we can really take an image like this, for example, and find all the objects of interest, in this case animals, put a bounding box around each one, tell you which species there are, and do even pose estimation, this stick figure thing, and tell you what they're doing, and even do environmental reconstruction off the shelf. Close. The thing is, the type of AI that is available off the shelf may not be the most appropriate type of tools for specific tasks of biodiversity monitoring, understanding, and mitigation. And the reason is that it hasn't been developed in the context of AI. So how do we know when can we trust off-the-shelf AI, and what kind of AI should we trust? So that's a pretty deep philosophical question of how do we engender trust in the first place. And a lot of the kind of, if you look at the literature or sort of the recommendations of trust that don't come from AI, but come from different disciplines that are either social sciences or business, the one of the first things, it's not about an accurate method, a good benchmarking, or any kind of other engineering measures. First and foremost, it has to be inclusive, a collaborative partnership between the people asking the questions, those who are intending to use the tools that are being developed, and the tool developers, so that the solutions are created with whom they benefit, not for them, right? And there has to be the ability to use the tools that are being developed. The tools need to be transparent. Um, and it's not just open source. There has to be explainability, or at least interpretability, which is a huge challenge in AI currently, with clear assumptions and uncertainty quantifications. And the responsibility for the outcomes, right? There has to be accountability. So AI safety is a big issue. And increasingly, because AI is now being used everywhere, not just for biodiversity monitoring, but this notion of do no harm translated into the context of AI, it has to be improvement on existing, including the cost of resources. Just because we can use the shiniest, latest model doesn't mean we should, right? There also has to be a protection of not just protection, but data governance and considerations of fairness and bias. And bias, as I pointed out, is not uh, is not just about sort of the demographic notions of bias, but the fundamental data distribution is biased. So going back, it's not just data that is biased by design. If we look at where AI is being developed, again, there's almost complementary kind of um, map of where majority of AI is being developed versus biodiversity hotspots, meaning that majority or almost all of the AI being developed in absolutely absence of context, of any kind of biodiversity context. And so it is not surprising then that when the conservation and more broadly biodiversity kind of sciences and practice communities is asked, surveyed about the use of different technologies, and this is from the Wild Labs community, which is the Tech for Conservation online community. And when asked about a variety of tools, AI consistently for the last 3 years comes up as at the bottom of the current performance, but at the top of the potential, right? So it's a tool that under-delivers in the context of biodiversity science and conservation. And so it's clearly we trust the potential, but it's not yet there to be used as a tool. The other challenge about using AI in biodiversity, particularly in the context of biodiversity and conservation, is that it's expensive. And conservation and biodiversity monitoring is already under-resourced. And it's not just resource, when we bring AI into the conversation, it's also about the resources of money, people, expertise, right? Tools and metrics. It's massive. AI is expensive in terms of the skill set and in terms of the people who can deliver on that skill set, as well as the actual natural resources of electricity, water, and the CO2 emissions. Increasingly, AI is becoming a drain on these resources, especially with a new with a new type of AI, the generative AI, and the big foundation models. Um, just the use of electricity today is at about 3% of total electricity use in the US, energy use in the US. By 2030, it's estimated to be 8% of all of US use would be by data centers and AI-related tasks. So, and it's not just for training these models anymore, for inference as well. And so increasingly the CO2 emissions are also the share of CO2 emissions associated with AI is increasing. And so if we're going to use AI to address biodiversity loss and mitigate the factors contributing, then we need to be aware of what kind of AI we're using and how we're using it. And so the other aspect, and finally, the last challenge is that AI currently is used primarily to speed up the monitoring, right, to fill the data gaps. But there is a huge opportunity to use AI-driven sort of policy coordination and evaluation of interventions. And so I'm going to show a couple of examples here. So all of these challenges and opportunities come from the Global Partnership on AI, which is now part of OECD.ai, which is Organization for Economic Cooperation and Development, published several reports on challenges and opportunities for AI. One of them is biodiversity. So I was part of the expert committee for this, outlining these challenges on data, understanding, resources, policy, and action, as well as the recommendations that connect very directly with the trustworthy, what makes AI trustworthy. So first of all, data need to be open, available, but protected where necessary. And so we really need to build a data infrastructure. The second part is that we need to prioritize the outreach and inclusion of local communities because biodiversity is a global problem with very, very local impact and local sourcing, right? So the biodiversity is very explicitly geolocated, and it's primarily within the lands and jurisdictions of different communities, most of it not federal government, even in the US. And certainly, generally doesn't respect, biodiversity doesn't respect the geopolitical jurisdictions. Funding, need to invest in actual both applications and cross-cutting digital infrastructure, and develop tools and metrics that help explicitly biodiversity monitoring and intervention evaluation, and finally, build capacity. And that is outside of the AI community, but in the biodiversity science and practice community. And so this is really only one aspect of it is we need better AI, which goes very much counter to the current kind of practice of the machine learning communities of how we develop and evaluate machine learning methods. So the current practice is that we build these stereotype benchmarks that everybody measures against, and the metrics that we use are accuracy, which is, again, very narrow set of metrics, and the results have to generalize across a variety of tasks, which does not work in applications, particularly if you're trying to figure out, I need, for example, metrics and indices to determine the conservation status of this particular species. One of them is population size estimates. I don't care on the accuracy of species classification or object classification overall, or even individual object identification overall, if it does not change my population size estimate, right? Moreover, every benchmark that's available out there is not going to be particularly useful when these methods are deployed in the real world, both because almost, as I showed already, almost all data sets are hugely skewed with these long tails where we have the bulk of data where we know a lot about very little and then almost nothing about the rest. So, and that means that if we're optimizing these metrics like accuracy, we're going to just get better and better about this very small part of data where we already know a lot. And so changing that mindset, here's one of the prime examples of a beautiful example of what is a good and trustworthy method that kind of brings it all together of AI and biodiversity is mega detector. And it's this very basic method for figuring out whether there is an object of interest, i.e., typically animal, in camera trap images. And so it was developed almost 10 years ago now, first by Sarah Beery, Dan Morris, and Xue Yang, um, to take a very simple model for detection, for object detection, which is not at all the type of models that are kind of currently developed by GPT or Anthropic or others. It's a model that can run and does run almost continuously on these tiny devices like your phone, or it can even smaller. So it's super lightweight, very few resources by comparison. And all it does in camera traps that are motion activated or infrared activated cameras, they we want them to activate when there is an animal in the image, but they are typically activated also when there is wind that moves the branches or a car passes by or human or anything else. And so you have to, typically we have about 70 to 90% of the images that are being taken, but those are blanks. They do not contain animals. So you want very quickly to filter out the images that contain animals or something of interest. And so the question was posed by the community that already uses camera traps as the primary source of biodiversity monitoring. The solution was designed, adapted from existing methods, lightweight, just good enough to this context, and it's been deployed on millions and millions and millions of images now. And it's still the primary use, the primary kind of method for biodiversity monitoring pipelines, particularly from camera traps. So it develops tools, it brings in massive amount of data that was inaccessible before, and it also is very usable, but with minimal training. So it brings in a community. A different example of kind of method for biodiversity monitoring and science and practice that perhaps uh, kind of expands it from a completely different scale, at completely different scales and perspective, is the new foundation model for the Tree of Life, BioClip. So this was published last June. And so to quickly fill this gap of, the ability for any species, for any image, to be able to quickly tell not just what species it is, but if we can tell if it's new species, or can we tell the genus? Can we tell at least to the family, and fill the taxonomic gaps and serve as a foundation for many other downstream tasks. So this is a model that was trained. This is the new type of model. So with a new type of architecture, super expensive, trained on 10 million images, 450,000 species. So almost everything that's available in the Encyclopedia of Life and part of GBIF. So this is essentially every available image that was well benchmarked at the time, and now building the second version of it. So the question is why, right? If you're going to build these kinds of models that are useful for a variety of tasks, particularly if you're going to fill in the gaps in data, you need something that serves as a good foundation. And if you're going to do this, you need to do it. You cannot afford to do it many times and sort of incrementally update it. So because these kinds of models take enormous amount of resources. So it took 11 months to make sure that the taxonomy for every image of those 10 million images was cleaned up and aligned. It took days to train this model. It's already being used, downloaded since June last year, downloaded and used more than 650,000 times worldwide for a variety of tasks. Everything from detecting new species not identified before, processing beetle collections from the National Ecological Observatory Network, and serving as the first pass uh, suggestion of species for tree species classification from satellite images, just to name a few. We are gearing up to train the second version of this. Again, doing it responsibly because it has to be qualitatively different. So it's not just, yes, it's going to have 200 million images. It's going to have cover pretty much everything that's available in Encyclopedia of Life, so about 500,000 species. So more images, not so much more species, but it also going to bring in the genome for species that are available, species description, and a lot more information to start serving as the model that people can start querying for scientific knowledge as well as functional knowledge, and maybe drive directly to deriving these biodiversity monitoring factor, biodiversity indices, to start understanding the why, if not, if at least to go beyond the filling the data gaps. And to understand the why, we really need interpretability. And so this is the first kind of interpretable method that really builds on top of these the new type of AI, machine learning models, transformer architecture that is able to not just tell you, oh, this is the species of bird, but this is the species of bird because, like, here are the parts that are relevant specifically for identifying it as this particular species. And it does take quite we have now sort of increasingly more biology grounded versions of it that really can not just tell you this is the species of bird or butterfly, here's why, but start separating between very, very similar looking species. So this is, believe it or not, the bottom left and right are two different species of Heliconius butterflies. And the highlighted areas, so automatically highlighted areas, point at the differences. So there's the very, very tip of the wing that's highlighted. And then the bottom, those magenta arrows are the ones that what the scientists develop as taxonomic keys, the kinds of binary trees, decision trees that separate between two closely similar looking species. So this point for on the left side, Heliconius melpomene does not have a white set of white spots on the wing at the top of the wing, while Heliconius auratus, which is Heliconius elevatus, Rato does have those white tips. And so we automatically detect these as well as this last white spot, which is different in the Melpomene versus Elevatus, and the shape of these rays on the bottom wing, and even suggesting potentially new differences, such as the curvature of the wing that's shown there. So we can start. And it matters not just because we can point out, oh, cool, we can now tell the difference between different species. We can start quantifying them. We can start quantifying within species diversity and start associating it and correlating it to different habitat conditions, and maybe start predicting of what will happen or which species may be vulnerable to the changing habitat conditions, right, to the changing environmental conditions. The other direction of using and developing AI methods is to really drive towards develop the indices that are relevant for conservation status assessment. So one of those is, for example, population size estimates, and the best way to do population size estimates are from individual ID, which is until we started developing until about 10 years ago, was missing really from the set of toolkits. So we could take these millions of images coming from all the sources. We can detect the ones that contain animals, for example, using mega detector. We can put a bounding box around them and identify species, perhaps using BioClip. And we can also go beyond the species, but ask not just, oh, this is a gravy zebra, but this is a gravy zebra because, sorry, this is Zippy the zebra, right? And we can even say because. And so this kind of individual ID, because it's the pattern of its stripes is the biometric unique identifier, just like every one of us has our own unique features that make us our own unique individual, from the color of our iris to the fingerprints to the shape of our ear, the sound of our voice, and the gait that each one of us has are unique to us. The visual, a lot of the biometric features are unique and unique to every animal. And this is not just zebra stripes, this is anything striped, spotted, wrinkled, notched in the water or on land, including also the shape of a whale's fluke or the dorsal fin of a dolphin. These are all unique. And so you can now imagine there's like tons of species that we can use these biometrics to identify. And so the question is, so what? Well, so what is that you can now build these platforms with data management for all of these species, and you can see the range, including my favorite is the sea dragons, the weedy and leafy sea dragons that look like aliens, but yes, they're uniquely identifiable. And then, because we can identify each individual, you can, with information on when and where the image was taken, you can start tracking an individual without putting GPS collars, but through through just images. You can count them. You can even do for social animals. You can do their social network. And so here's an example of a platform that we built, Wildbook, which is the platform of the nonprofit WildMe that was mentioned, which is now part of Conservation X Labs. And this is Wildbook for sharks. Each page here, so this is a particular shark, MXA 700, which is a Mexican. So this male hangs out around Cancun, mostly around July and August. And you can see all the different observations that were made of this specific individuals by different people throughout the years, including some that were posted on YouTube. And an agent, autonomous agent, AI agent, noticed and automatically added. At the bottom of each page, we also list everybody who contributed information to this specific individual. So it's not, these are global species. So there's not one project, not one organization that has information even about one individual whale shark. These are whale sharks. It's just, but if we put together the information from all the organizations, from everybody who has observed, we can actually put a global picture of a global species. These sharks travel more than 5,000 miles, massive migrations. And so the Wild Book for Sharks has contributions for more than 22,000 identified individual sharks that come from scientists, conservation organizations, and even volunteer contributors. Massive data set now that is shared across the globe And because it is shared, because people contribute, because they themselves contribute data, they trust, not only trust the data that's there, they trust the sharing of the data. There is the incentive to share. We'll list these, the contributors to this particular shark. They know already now that the information that they may need, somebody else might have it. And so it incentivizes sharing and community participation. It also reduces the cost of data compilation and the tools that are built on top of it. And so with that shared data, it is that data in Wildbook for sharks that became the foundation for the species assessment for whale sharks in the International Union for Conservation of Nature Red List, ASEAN Red List. So the species were reassessed in 2016 based on that data, and the conservation status changed from vulnerable to endangered, and the population trend from stable to decreasing, not because the species are doing worse, but because we have better data. And based on that data, more complete, less biased, more trustworthy, we're able to make better decisions, better assessment that lead to better resource allocation, because the moment that the species status goes from vulnerable to endangered, it triggers completely different set of policies and resource allocation. It also, because the data are trustworthy, and it's built on a pipeline of machine learning and AI methods that are driven to provide not the abstract notion of accuracy, but the ability to have accurate population size estimates on top of it, and tracking individual animals and a whole bunch of other statistics, that the best understanding, the most comprehensive study of the species was published using that data and co-authored by 37 authors that met through these pages of Wildbook, where at the bottom that's listed who contributed to that data to this particular individual. And so this is an example of AI that's used not just to understand biodiversity and the seasonality of migration patterns of these whale sharks, so you know where and when you need to put resources to protect their habitat, but also and support policy and action and engage communities and trust. The same technology, and I'm wrapping up, the same technology was also used to enable the first ever full census of an entire species. This is the endangered gravy zebra, so this is terrestrial species now, using just photograph from ordinary people just driving around the country, Kenya, for 2 days. For the first time, it was in 2016, in January. Hundreds of people were driving around Kenya taking pictures of every gravy zebra that they saw, and 95% of all gravy zebras, this endangered species, are in Kenya. And these are not the zebras you're thinking about. The ones, the 1,000 Serengeti migration with wildebeest, those are plain zebras, not these ones. There's only, and so in 2016, after hundreds of people took about more than 40,000 pictures of these species, and by hundreds of people, I mean truly everybody from school kids and local park rangers to tourists with telephoto cameras, and even at the U.S. ambassador to Kenya at the time, here, Bob Godek is there in this picture up in the center. Because we're able to identify every individual zebra, we're able to provide very, very accurate statistics, statistical estimate on the population size using marker capture method, showing that the population size was 2352 plus minus 72, tighter bounds than any other method at the time. So good, the confidence bounds, that Kenya Wildlife Service went complete 180 from this is not how we do a population size estimate to this is exactly how we're going to do it, and you're on the hook every 2 years. And so the whole effort was repeated in 2018, more people, more locations, more pictures, and turns out more zebras. So that was, the population size was 100 plus minus 150, again, because of the uh, tight confidence bounds that the decision makers were able to trust, they could truly say, yes, the population is growing. And there are interventions that were made based on this. And the foundation of that trust was not just a good algorithm, but the fact that we got donations of about 200 GPS-enabled cameras, that we came up with a very old school way of making sure that everybody, even if you don't have a GPS enabled camera, was able to synchronize their data to something that was GPS enabled using this very simple kind of cards. Working with local nonprofits, many nonprofit organizations who created a whole kind of guide to the whole event, what's going to happen, how to take great pictures, why are we doing it, what organizations are participating, and even sort of driving some competition, and went around the country explaining all of it, not just in English, but in Swahili, in Maasai, so that everybody could participate. And we also trained representatives from different nonprofit organizations to use the system so that they could actually we wouldn't be the bottleneck. Okay, it took a couple of times. We're still training, but building that capacity so that people can use the system and do this analysis for themselves. And it is because of that that the Kenya Wildlife Service and all 6 governors of the counties that have gravy zebras came together and changed and came up with a whole endangered species management policy and proclamation that puts money, resources, people, policies to protect the species based on this data, because the tagline for that event was Kenyans powering conservation, because the data came from the community, the analysis was done with the community, and the results had meaning for the community. And so this, Simon Gitau, the associate director for Kenya Wildlife Service, said this shows the power of citizen science and machine learning for conservation. The IUCN Red List entry for gravy zebras was also changed. And so this is an example of AI to understand biodiversity drivers, and support policy and biodiversity action, because there's trust at every step. And so hopefully this is kind of a taste and a couple of examples of how we not only think about trust by design, but also what it can enable, and how we can truly meet these challenges of data, understanding, resources, and action. Of course, I'm standing here, but the work is done by many people, not just the Imageomics Institute and the AI and Biodiversity Change Global Center and the Wild Me and Conservation X nonprofits, but many years of collaborations, direct collaborations, as well as the people who ultimately go out in the field and use these tools and ask the questions and drive the change that is actually necessary in the field. So, because when we together succeed, the payoff is massive. And the benefits are not just to, as I said, the nice to have, it's the need to have. And hopefully we'll get there. Thank you.
Thank you so much, Tanya, for such an excellent and insightful talk. Um, you can't hear, but from our lounge, there was a great round of applause from everybody. But now I'd like to open it up to the audience to start our moderated Q&A portion. So please feel free to use the Q&A button to type your questions regarding the talk, and we'll be able to see those questions on our end. And to start us off, I thought that the Kenyan Wildlife Service and the zebras example you gave was a really interesting example of how you could bring in local communities and local contexts and partners. I'm curious to learn about the challenges uh, and opportunities that you see in encouraging more citizen scientists to participate in uploading and sharing their photos with your projects?
Yeah, so I mean, like everything else, citizen science and sort of more crowdsourced contributions are they're cons and pros. So it is not a coincidence that the current existing citizen science platforms are very biased towards North America and Western Europe, or well-resourced locations. To begin with, quite often, you need something like this, right, to be able to take. That said, that the the devices are increasingly available almost everywhere in the world. What's not available, and so majority of contributions, citizen science contributions, come from where people are, right? So then the incentive is, how do we get people where biodiversity is, and how do we do it responsibly? So on the one hand, majority of biodiversity is not where people are. It is in hard to reach locations, and maybe should be not reached locations. We've reached enough locations with results that maybe not are not the best for biodiversity. So creating technology that has super light footprint while engaging people, it's a hard balance. It's a really a big challenge, which we, in fact, we're just about to put out a policy document from the Computing Research Association's Computing Consortium on this intersection of computing and citizen science, of where the opportunities are. And one of the big ones is this light, but very accessible infrastructure, right?
Okay.
Yeah. And so the other aspect of it is, and the balance, oh, there are questions now. And the balance is, what's gold for science and conservation management is also, unfortunately, super useful for wildlife crime and wildlife criminals. So understanding sort of how we protect that data and where data leakage is happening is also, as the bigger collections of data we're creating, the easier it is to extract patterns out of it, which is what we want for science. But it also makes the location of, especially endangered species, more predictable. So we need we need to do it responsibly as well.
Great. Thank you. Looks like we have a question from Ranim asking uh, if you could speak more to how the pictures of the zebra were monitored for repeated records of the same individuals.
Um, sorry. So the individual ID, how do we do individual ID?
Yeah, I believe that was the question.
Yes. So there is now a variety of methods to do this. Let me kind of talk about zebra stripes maybe. So if you stare at it long enough, you'll start realizing that the pattern is different between different zebras. And so what's easy for humans is we can answer same or not same. I can tell you that if you stare at this for even a couple of minutes, you'll start seeing the differences. So if I show you two pictures of zebras, you can actually tell same or not same. Is it the same individuals or not? But with computational approaches, we can really now start building methods that are both based on straight up computer vision. So this is algebra, actually very simple piece of algebra, but very clever one that kind of creates a signature of not the pixel values, but the changes in the pixel values, the transition of black to white, white to black, or like spots and sharks. So anything striped and spotted can be kind of, you can create this kind of signature, which very quickly matches, allows them to match one image to another. And we can extend these methods um, to sort of more deep learning kind of methods that do this a little faster, and while still providing the ability to say, here's why. Like these are two, this is the same individual, not the same individual. Here's the matching part. If I'm quick enough, I may be able to come back to this and give you concrete examples of the computer vision pipeline if that's of interest.
Oh,
Um, yeah,
thank you.
but the point being here, it's a very, very challenging computer vision problem or image analysis problem because it's very easy if you're doing matching. So if you have like, here's my total 3,000 zebras, and I know I'll never see another zebra ever again. So then any new image of a zebra I can match to an existing one. But the thing is, what happens, like everything else I said, you get this very, very skewed distribution. For all the species that we have, about half of the individuals have only one image, only one photo, which means that majority of the query is not which one of the existing zebras is this one, but do I have this one already? Have I seen this one already or not? Or is it a new one? And majority of the answer is yes, new one. And I need to be able to tell that confidently. And so a lot of the kind of underlying computational machinery goes into this ability to provide a confident answer that it's a new one.
Interesting.
Mhm.
Um, Looks like we have a question from Art in the Q&A also, if you can see that.
Yeah,
Um,
so there are two good questions here. So the first one is how can we make sure certain that AI does not come with its own biases? It's a great question. And so um, the biggest source of bias right now for most of the machine learning methods, that especially the ones created here, are the bias of the data that they're being trained on, right? And so for different contexts, for different tasks, for different data sources, there are different ways of guarding a little bit against this bias. But one of the first ones is this recognition that we're not creating yet another machine learning. The point of all of this is not to create yet another machine learning method. The point of it is to create tools that scientists and decision makers can use confidently, right, for the questions and the tasks that they need to use. So, for example, if we're staying with this estimating population size, the goal is not to create more accurate method for, the most accurate method for um, individual individual zebra recognition and to put all the resources in that. If your population size doesn't change from the current method that, let's say, I give you to something that, let's say, I'm able to improve my computer vision method to from let's say 87% accuracy to 93% accuracy by massive increase in resources. Should we do it, right? And the answer is probably not, right? What we should really focus on is providing the ability for the human curation, for easy human curation, right? And knowing when to trust the current machine learning results, computer vision results, computational results versus when to ask a human. And so we think quite often in these kinds of trade-offs, not to drive. So yes, let's get better data. That's one way to debias things. Let's let's invest some amount of resources into building these general purpose massive foundation models that can then be used by everybody else to build specialized tasks. But then also thinking about as a partnership between machine and human, where we measure the performance of the system, not just the accuracy of the computational side of it, but the confidence, the trade-off of where we trust the computational side and how do we decide what should be curated by humans or answered by humans, in the improvement on the decision ability of humans to make decisions confidently or to um, do new science confidently with the amount of effort that it takes, right? So until now, let's say humans had to look at every zebra, and they still made about 10, 15% error. We now verified this. And the confidence bounds because of that were pretty wide with the current methods, ways of estimating population size. And so the decisions that people could make based on that data were very different that when we narrow this confidence bounds. So now we have a whole machine learning pipeline that reduces the human effort to maybe about 5% curation, right? But it increases confidence bound, it tightens the confidence bounds, and while maybe staying within the same error bound, probably better than the. So it changes the types of decisions that that are being made on a much better, hopefully, data. So this is an example of how we think about sort of biases. Biases are there no matter what. We mitigate what we can, and then think of it as a partnership.
Thank you. That was an interesting way to think about the trade-off. We are at the hour now. Looks like there were a couple more questions.
Yeah, I'm happy to answer those, and maybe taking less time to answer those if that is an option.
Sure, yeah, we could do maybe one more question from the Q&A.
Okay. So thank you for the reef check. Happy to follow up, probably separately. That's a great example. And then the question on how well the species estimation correlate with other factors and more traditional methods, and what do we do for invertebrates? So those are great questions. So so Amy collaborated, David Rolnick and several others in the AI and Biodiversity Change ABC Global Center, building special camera traps for moths. So this is really focusing on invertebrates. So camera traps that attract moths, and then the whole machine learning pipeline that detects, identifies the species, and even suggests new species. We also have a massive project for beetles leveraging the entirety of National Ecological Observatory collection. So this is really going beyond vertebrates and really, really focusing on invertebrates and fungi and plants. So working with the Smithsonian Institution and others for using scan herbaria collections. This is actually the massive source of plant images and sort of filling our taxonomic gaps. And then about the correlation, there's a great question about correlating different sort of observed metrics and observed maybe traits and morphological traits or patterns to and indices, computed indices to other ecosystem measures from OpenSet. So this is another example, and I'm going to try to very quickly. One of the first data sets that really this multimodal is the answer, and it's actually probably the answer to many things, including every modality is biased. The only way to get something is maybe to put all of these modalities together. And so the one of the nice examples, more recent examples of this is the new data set that just uh, came out on trees. And that brings together everything from very individual tree like circumference and tomography measurements all the way to satellites. And so really starting to learn how to correlate across different modalities, different scales, um, and not just build the technology side of it, the toolkit side of it, but also the science of it to really scale, to increase our scale across time, space, and individuals, while also increasing the resolution of our sort of object of study.
Awesome. Well, we are at the hour. So unfortunately we couldn't get to every question in the Q&A. But I want to wrap up by thanking you again for such a fascinating talk and conversation. Thank you so much for your presentation. And to everyone online, thank you for joining us in this first talk of the seminar series. Please join us again on May 23rd and June 25th for our next two talks by Victoria Nguyen and Bonnie Lei.
And thank
Thank,
you again. And if there are more questions, please reach out to me on email or contact ABC and Biodiversity Change Global Center, and we'll be happy to follow up.
Great. Thank you.